Hello, my name is Tyler Funnell. I'm a fish biologist with the U.S. Geological Survey Great Lakes Science Center, stationed out of Hammond Bay. Today, I will be giving a project update on this project, the habitat use and movement patterns across the Lake Whitefish stock complex in Lake Huron. This is a parallel project to the one just presented on for fish tagged in the Canadian waters. Today, I will be talking about fish tagged in the U.S. waters of Lake Huron. And this project is still very much ongoing, and the data are still preliminary. So this is just to give you all a snapshot of the data that we're seeing come in for this project, and give you an idea of the direction that we're headed with this project moving forward. Before I get started, I would like to acknowledge the long list of folks on this list that have contributed to this project, both the research team and field crews out there in the field who are tending receivers and tagging fish. All the people on this list have made this research possible, so a big thank you to them. And I'd like to acknowledge that the funding for this project is through the Great Lakes Fishery Commission. Lake Whitefish and Lake Huron are undergoing declines in recruitment and commercial fishery yield in Lake Huron. This figure here shows commercial fishery yield over time. And what we can see is that there was a peak in about the year 2000 and a steady decline since then. And a lot of research effort has focused on the mismatch in recruitment that we're seeing and some potential drivers behind low recruitment. But what we're doing in this study is trying to take a look at things from the adult perspective and see how the adults are responding to potential ecosystem changes and what that might mean for management. And the ecosystem in Lake Huron, like in much of the Great Lakes, has undergone drastic changes in recent years, particularly due to dreissynid mussels, that's zebra and quagga mussels, their invasion and proliferation, and subsequent invasions of species like round goby, which are egg predators to species like lake whitefish. And these ecosystem changes have also led to the collapse of an important prey item, Diporia, this freshwater shrimp you can see on your right. And we have a lack of fishery independent movement data from adults at a whole lake scale. So these figures here show some work done by Mark Ebner and others back in the early 2000s, which involved mark recapture methods. So this involves just collecting two data points on fish, where they're marked and originally caught, and then where they're recaught and reported by fishers. However, this relies on fish only being reported as recaptured in areas where there is fishing effort. So what we try to do in this study is take this a step further using some fishery independent methods. And in doing this, we ask three research questions that we hope to begin to address through this presentation. The first being, what is the potential for mixing among spawning populations, particularly during the non-spawning season? How have movements of lake whitefish changed over time since that Mark Ebner study? And what source sink dynamics may exist in this system? And those source sink dynamics meaning that if fish are produced in one area and then are captured in another area, what are the dynamics going on there? And how can management respond to address the mismatch that's occurring in recruitment and capture? And to answer these questions, we use acoustic telemetry technology, which involves tagging fish with an acoustic transmitter, this blue icon in the belly of this fish, and releasing that fish into the lake where it can be detected on acoustic receivers, which are anchored to the lake bottom all over the lake. And these pictures below just show some of the tagging efforts that went on. And these are here to indicate just what a large scale effort this was. And once again, want to say a big thank you to all the folks that were able to come out and help tag. It took an army in this case, and I'm very grateful to all the support that we had in the tagging efforts. Our lake whitefish were tagged across three general regions in the U.S. waters of Lake Huron. These were in the north, in the central region, and in Saginaw Bay. So these north tagging events occurred off the ports of Hessel and Detour. The central region were just inside of Thunder Bay and just around to the north of North Point here, north of Thunder Bay. And then we had two tagging locations here in Saginaw Bay. We tagged fish out of Tawas and out of Caseville. These fish were all tagged in what we consider to be the pre-spawn period. So this was October or very early November. And because of this, we assumed that these fish were of somewhat of a mixed stock, but that fish were likely beginning their migrations back to spawning grounds. So we're likely tagging fish from a general spawning region, but we weren't picking fish off of their direct spawning grounds. And in total so far, on the US side, we have tagged just under 650 lake whitefish, and we will be tagging about 150 more this spring if we can get out. So today I will be providing some preliminary observations. Like I've mentioned, this project is ongoing, so we will just be taking a look at information from fish that were tagged in 2022 and 2023, with the detection period being from the time that they were tagged through to June of last year. And every summer when we go out and we pick up these receivers, we get another year of data from these fish. So we'll get another big batch of data this summer when we can get out there and collect those receivers. So what we can see here is on this map, we see, again, those tagging locations from the fish in 22 and 23, and each of those black dots represents an acoustic receiver. So a fish could have been heard on any of those acoustic receivers. And each of those with these tags have a detection range of somewhere around half a kilometer. And what we are looking at today are first the overall space use for each of these tagging groups. Second, we'll be touching on a bit of a historical comparison. And then third, talk about how these fish behaviors are corresponding to management units. And all this, like I have mentioned, is just scratching the surface. We are still collecting a lot of data on these fish, and we haven't even tagged all of our fish yet. So these are by no means final results or takeaways. These are just the early trends of what we're seeing from these fish. So getting into some results. This is to familiarize you to this figure. I'll be showing these on quite a few of the upcoming slides. So what we're doing here is breaking down fish by their tagging location. So these fish were tagged in Hessel, which is indicated by this red star. And what we see on this figure is any of the colored circles are acoustic receivers where at least one fish was detected. And an X indicates an acoustic receiver that could have heard fish, but it did not hear any fish from this tagging group during this season. So this first top left plot shows the fall season from October to December. And the color of those points indicates the number of fish that was detected on one of these receivers, with the darker, more purple colors indicating the most fish, and yellow indicating only a couple fish. So our big takeaway here from October and December for these fish tagged in Hessel is that the majority of these fish, the darker colors, are showing up right around where they were tagged, which makes sense because they were tagged in October. But you can see some of these fish moving across the coast, the UP coastline here, and up through Detour Passage. Pulling up the rest of the seasons here, we have winter over here on the top right, we have spring down here on the bottom left, and summer in the bottom right that we'll only have for some of these tagging groups, those that were tagged in 2022, which happens to be Hessel. But what we see, some general takeaways here, are that as we get out of fall and into the other seasons, we see two movement groups kind of coming about from these fish. So one clump of fish is hanging out right around where they were tagged in the Lachino Islands area here, some moving over toward Mackinac Island. And another group of fish moved over to Detour Passage, some moving through and up into Pottagenesing Bay and the very western edge of the North Channel. And this trend holds coming into summer, with fish leaving Pottagenesing Bay since it's shallow and occupying either Detour or along this UP shoreline. So a couple fish did go all the way down to Outer Saginaw Bay, but the vast majority of fish stayed very close to where they were tagged. Fish tagged in Detour here were much of a similar story. We see during fall, those fish are right there tight, close to where they were tagged, with some movement to the north happening up into the lower St. Mary's River. And then pulling up the other seasons here, we see something very similar to what we see in fall, and that is that fish are staying very close to home generally, staying near that Detour Passage area and just poking their head up into Pottagenesing Bay and some into the far western edge of the North Channel. But once again, fish are staying very close to home with this tagging group. Our next tagging group, we're moving south and looking at fish that were tagged near the port of Alpena, including those tagged just outside of Thunder Bay, as well as some tagged inside of Thunder Bay. And we can see already that these fish are behaving a little bit differently and that they are moving much longer distances. Even just during this first season after they were tagged, fish are moving down into the Saginaw Bay area and the southern main basin of Lake Huron. And pulling up winter and spring, we see those trends hold. Fish seem to be mostly vacating the area in which they were tagged near Alpena and generally moving south, or exclusively moving south, I should say, into outer Saginaw Bay and southern Lake Huron. So we kind of see two movement groups coming about here. That's pretty clear when we look at spring. There's this nice dark clump here in the outer Saginaw Bay, and then another clump down in the southern Main Basin. Moving next into East Tawas and Caseville, so our two tagging groups that were in Saginaw Bay. We see first in October and December that we have some fish detected all over, but again, remember that those yellow dots just mean that was one or two fish detected there. So our darker dots that we're most interested in are detected right in Saginaw Bay, with a good number of fish spilling out into the main basin to the south, and just a handful of fish moving to the north. And pulling up the rest of the seasons here, we see a lot of that same trend that we saw from our fish tagged up near Alpena, and that is that we have two movement groups forming. Fish that are hanging out in Saginaw Bay and fish that are hanging out in the southern Main Basin. And if we move down and take a look at summer in the bottom right here, unsurprisingly, we can see these fish vacating inner Saginaw Bay and just holding up outside in deeper water in the outer bay here. And again, we have fish in the southern Main Basin as well during summer. So now we're taking and combining all of what I just showed you from each tagging location. So rather than breaking it down by season, now we just combine all seasons here for this plot on the right. Once again, the star indicates the tagging location. The dark points indicate where there were the majority of fish, and those yellow points just indicate a couple fish. And the X's mean that there was a receiver there, but no fish were detected. The new figure to us is the one on the left, and that shows some mark recapture data that span from 2003 to 2012, and this was work done by Ebner and others. The circle shows the tagging locations, so we can see the tagging locations are very close, and we're just comparing over time here. But what this plot over here shows is that the darker colors indicate that more recaptures were occurring in that grid cell, and lighter colors indicate that fewer recaptures were occurring. So generally what we see here, if we compare the left to the right, is that we saw similar behaviors across this group of fish that was tagged around the Les Chenaux Islands area, in that the vast majority of fish were detected along that UP shoreline and stayed close to home. But there were some stragglers in both studies that poked their heads into the North Channel or made their way down south. But again, the vast majority of fish staying close to home. Next, looking at the Alpena area, we see some differences between that plot on the left and the plot on the right. And the main difference is that if you look at today's data, the acoustic telemetry on your right, there are no detections north of where this map is cut off. Whereas if we look at the mark recapture data, we see that fish are moving north and they're moving south, with some detections along the UP shoreline and Manitoulin Island, as well as down in Saginaw Bay and the southern Main Basin. So what this means is that in our acoustic telemetry data from this study, we seem to be missing that northern movement group. Whether or not we just didn't detect it, we didn't tag fish belonging to that group or that exhibit that behavior, or if that behavior is less prevalent now than it was then, we're not sure, but we're hoping to get some clarity on that question here in the coming years as we tag more fish. And a somewhat similar story here in Saginaw Bay. We see that for these fish tagged by Mark Ebner in the early 2000s, that some fish were going all the way up to the UP shoreline in Manitoulin Island, but some fish were staying close in Saginaw Bay and the southern Main Basin. And in our data from the acoustic telemetry study that I'm presenting on, we saw just the Saginaw Bay, southern Main Basin, and a couple fish nosing up to like the Thunder Bay area, but no fish moving further north than that. So again, a difference there. Moving now into the last results slide, we can see here a histogram showing the number of different management units that fish were detected in. We can see that the majority of fish were detected in 2 or 3 management units, with some even being detected in up to 7 different management units. And ultimately, 85% of fish in this study were detected in more than 1 management unit. And this is generally similar across the different tagging locations. Anywhere from 75% to 100% of fish were detected in more than 1 management unit. So the fish are not necessarily sticking to the invisible lines on the map. So future work in this study will expand on how the fish are using these different management units. So some quick takeaways as I wrap up here. We see a high degree of mixing between the fish tagged in the Alpena area and fish tagged in the Saginaw Bay area, but nearly no mixing between those 2 tagging groups tagged in the UP and the different tagging groups tagged along the Lower Peninsula shoreline. And looking back to the historic comparison, those fish tagged around Hessel and the Les Chenaux Islands area behave similarly to those historically, but in Alpena and the Saginaw Bay, we saw different behaviors now than we did 20 years ago. And many fish are using multiple management units. And on all of this, like I've mentioned, there's a lot more to come. So I'm excited to share that in the future. And with that, I'd like to say thank you so much for your time and attention and the opportunity to present today. I wish I could be there in person, but please feel free to send me any questions or comments that you might have. My email is there, tfunnel@usgs.gov. I look forward to hearing from you. Thank you very much.